Welcome to the deep dive. We are jumping straight into the sources you shared with us, and today we're tracking this convergence of three really monumental shifts in science.
Mm.
It's how we measure aging, how AI finds new drugs, and then how the latest pharmaceuticals are. Well, they're fundamentally redefining metabolic health. This is the very near future of medicine, and it's happening right now.
It is. It's a truly exciting phase. For the longest time, the science of longevity was, mostly correlational.
Right, A is associated with B.
Exactly. And our sources today show we've moved into an era of causality. We're not just looking at markers that go up when you get old. We are using technology to identify the molecular changes that actively cause the aging process.
And that's our mission today. We're going to unpack these three interconnected threads. So first, we're going to look at the new gold standard in metrics. These causality-enriched aging clocks that can separate actual damage from, say, adaptation. Second, we're diving into this massive AI-powered discovery engine. It's already flagged surprising new anti-aging compounds and, maybe more shockingly, revealed some hidden risks in existing drugs.
And finally, we'll get into the profound implications of drugs like GLP-1 agonists, not just for weight loss, but for deep cardiovascular protection and a critical biological concept known as metabolic adaptation.
It's this convergence, right? Better measurement, scale discovery, and targeted intervention.
That's it. And that's why biological aging is no longer just a correlation. It's becoming a targetable medical process.
Let's start with the ruler then. For years, if you followed longevity research, you knew about epigenetic clocks. Horvath, Grimmage, they were everywhere.
They were revolutionary for their time.
But they were ultimately correlational. They told you what markers tracked with aging. So what happens when researchers demand proof? A true measure of causality.
Well, they had to build a far more sophisticated ruler. One of the sources details this process, and it relies on a massive computational technique. It's called epigenome-wide Mendelian randomization, or EWMR.
Okay, Mendelian randomization. So you're using genetics as a sort of natural experiment, right?
That's a great way to put it. Think of it as a tool that lets scientists use the random genetic variation you inherit to mimic a perfect randomized clinical trial.
So instead of just finding markers that show up in aging, they're using genetics to see which of these little chemical switches on our DNA actually cause an outcome, like frailty or a shorter lifespan.
Precisely. They analyzed genetic data tied to over 420,000 different CPG sites across the human genome. And a CPG site, for you listening, is basically a tiny dimmer switch on your DNA that controls how a gene is expressed.
And this is where the famous clocks, the ones we all know, they kind of fell short.
They did. This was the critical insight. When the researchers tested the most popular clocks, PhenoAge, Grimmage, even Dundon Pace, against these new causal CPG sites, they found a huge disconnect.
What do you mean by a disconnect?
The old clocks, they weren't significantly enriched for these causal markers. It suggests they were overwhelmingly measuring aging byproducts.
The consequences of the damage.
Exactly, the consequences, not the fundamental molecular causes.
Wow. So an intervention could make your Grimmage number look fantastic. But if it's not actually hitting those true causal sites, you might just be papering over the cracks.
You might be improving a side effect while the underlying damage is just accelerating. It makes you wonder about a lot of studies from the last 10 years.
It absolutely does.
And this realization, it led to what they call the big split in modeling. They created new causality-enriched clocks like damage and adaptage.
Okay, damage and adaptage. What's the difference?
They were built specifically to differentiate between two kinds of methylation changes. You have the damaging changes, which drive the bad stuff, and then you have the protective or adaptive changes, which is your body fighting back or adjusting to stress.
So now we can actually tell if an intervention is repairing damage or just making the body work harder to cope. That's a fundamentally different approach.
It's a huge shift. And, once you have a better target, the search for solutions can really begin.
Which brings us to the ClockBase agent platform. This enormous AI-powered system designed to just mine the entire history of molecular research for anti-aging clues.
I mean, enormous is an understatement.
Okay, let's unpack the sheer scale of this project.
They built an engine that harmonizes and reanalyzes. Get this. Approximately 2 million human and mouse molecular samples.
2 million.
Think about that volume. Data collected over decades for, cancer, metabolism, everything. And the AI applies the lens of over 40 integrated aging clocks, including the new causal ones, to every single sample.
That's data integration on a scale I don't think we've ever seen before. It's making all of molecular history legible for longevity research.
And it does it with remarkable rigor. It's a multi-agent AI framework, so it autonomously generates hypotheses, performs the statistical analyses, and then synthesizes the findings into these reports with composite scores.
And this isn't just a language model guessing. They actually validated it.
They did. The agent's analytical accuracy was 99.27% correct. It's doing real science.
That level of rigor is what lets us trust the patterns it found. So,
Yeah.
after analyzing over 43,000 comparisons, what was the biggest takeaway? How easy is it to reverse aging?
It exposed what they call a fundamental biological asymmetry. The data showed that significantly more interventions accelerate aging than decelerate it.
So it's easier to break things than to fix them.
Much easier. Disruption is biologically far more common than improvement. And, no surprise, disease states showed the highest rate of acceleration. About 24% of those comparisons showed significant pro-aging effects.
That makes intuitive sense. Disease makes you older. But the finding about existing FDA-approved drugs, that one stopped me in my tracks.
That is the crucial and somewhat concerning public health implication here. The overall skew suggests many of our current therapeutics, while they're essential for treating one specific thing, may carry a biological cost long-term.
What was the ratio?
The AI found a really stark one. 136 FDA-approved drugs showed pro-aging effects versus only 78 showing anti-aging effects.
That's a finding that needs to be shouted from the rooftops. We have this huge population taking drugs developed without any of this kind of analysis. And it looks like a lot of them might be speeding up biological decline.
Exactly. It highlights the need to rescreen basically the entire pharmacological landscape with these new causal metrics.
But it wasn't all bad news. The platform also showed its power for discovery, right?
It absolutely did. Amidst all those results, the AI flagged a really unexpected candidate for anti-aging.
An old drug given a new life.
Pretty much. The AI identified ouabain, a cardiac glycoside, as a top-scoring intervention. They followed up with in vivo mouse models, and the results were compelling. Intermittent ouabain treatment in aged mice led to a significantly lower frailty index and preserved physical parameters over 3 months.
That is hypothesis-free discovery at its absolute best.
It really is, using pure computational power to find new roles for old molecules.
That's revolutionary. Okay, let's pivot to a drug that is already well known in longevity circles. Metformin. You provided some really compelling data from a 40-month study in aged cynomolgus monkeys.
Right. When we talk about high-level validation, this primate data is about as good as it gets outside of direct human trials.
And these weren't young monkeys either.
No, this was a critical long-term study using monkeys that were already old. We're talking the equivalent of about 40 to 50 human years. The goal was to see if metformin could produce systemic protective effects, beyond its role in blood sugar.
And what did this multi-year study find? Can it slow aging across the whole body?
The findings suggest it absolutely has the capacity to ameliorate aging across the primate body. But what was fascinating was the mechanism.
That wasn't what we thought.
Well, we often link metformin to the AMPK pathway for metabolism. But the study pinpointed a specific non-metabolic mechanism that seemed to be responsible for the systemic rejuvenation they saw.
So we're talking about a kind of beneficial side effect that might actually be the main event for longevity.
Precisely. They found a deceleration of neuronal senescence and brain aging, and they attributed this primarily to the activation of the NRF2 pathway.
NRF2, remind us what that is.
Think of NRF2 as the cell's internal firefighter. It's an intrinsic antioxidant defense system that coordinates the cell's response to stress and damage.
Wait, so the anti-aging effect isn't necessarily coming from the gut or the liver, but from this deeper cellular protection, this internal firefighter starting in the brain.
That's the key takeaway. They identified it as a cell-autonomous effect, meaning it happens inside the cell no matter what's going on around it. It highlights a really potent systemic mechanism for rejuvenation.
Which gives us a lot more confidence that metformin's benefits are real, and they extend far beyond just diabetes management.
Exactly.
That brings us perfectly to the biggest pharmaceutical story in, recent memory. GLP-1 agonists like semaglutide and tirzepatide.
Absolutely.
The public focus is almost entirely on weight loss. But the sources you provided confirm there are profound implications for metabolic and cardiovascular health.
Let's start with the heart protection evidence.
The clinical picture is very strong. A large database study, one that was benchmarked against established trials, showed major cardiovascular benefits. Specifically, semaglutide lowered the 1-year risk of MACE.
Major adverse cardiovascular events, so heart attack or stroke.
Right. It lowered that risk by a significant margin, a hazard ratio of 0.82 compared to older diabetes drugs.
That's a real-world risk reduction that could translate to millions of people.
It does. And tirzepatide, the dual agonist, showed comparable benefits. The confidence in these findings is high because the study was designed to emulate established randomized trials, and the results matched up.
It confirms these drugs are fundamentally changing the risk profile for people with metabolic disease.
They are.
But here's where it gets, for me, really interesting. Moving past those clinical endpoints to the actual physiological mechanism, this concept of metabolic adaptation.
If you've ever tried to diet, you know your body fights back. So let's define the terms. What's RMR and what's adaptive thermogenesis?
Okay, so your resting metabolic rate, or RMR, is just the number of calories your body burns at complete rest, just to keep the lights on. Metabolic adaptation, or adaptive thermogenesis, is when the body slows that RMR down more than predicted based on how much weight you've lost.
It's an evolutionary defense mechanism.
A hyper-responsive one. Your body senses major calorie restriction and just screams, "We're starving! Emergency mode! Conserve every calorie!"
The classic heartbreaking example of this is the Biggest Loser study, right?
Exactly. In that study, participants lost extreme amounts of weight, but their RMR dropped by a staggering 500 calories beyond what was predicted.
Which is why so many of them regained the weight. Their engine was permanently throttled down.
It was. So the critical test for these new drugs is, are they just repeating that cycle?
Right. How did the tirzepatide study address this metabolic pushback?
This is the real aha moment from the 2025 data. Participants achieved a huge 15% body weight loss. But crucially, their resting metabolic rate drop was only as predicted from the normal loss of tissue.
So they didn't have that extra drop-off.
They did not exhibit the hyper-responsive drop-off. No metabolic adaptation.
Their body just maintained the metabolic rate it should have for their new lower weight. It didn't slam on the brakes.
It didn't slam the brakes.
What does this mean for someone trying to maintain their health long-term?
It means that tirzepatide appears to fundamentally protect your metabolic health during weight loss. It helps maintain a functional RMR. You see less muscle loss. You get increased fat oxidation. It breaks that brutal cycle where the body fights back.
So it offers a path to long-term weight maintenance that was, well, it was basically physiologically unavailable for most people before.
That's right.
What an extraordinary deep dive. We started with the very foundation, right?
Yeah.
Refining our measurement of biological damage with these causality-enriched clocks.
Mm.
We saw AI platforms just accelerating discovery, bringing us things like ouabain and confirming rejuvenation pathways for old drugs like metformin.
And then we end with these powerful metabolic drugs like tirzepatide, offering physiological protection against the body's own natural pushback.
This convergence is redefining our entire understanding of health span.
It really is. We're moving from observing aging to actively modulating it. And the knowledge from these sources is truly revolutionary for managing your own health trajectory.
Before we close, I want to loop back to that startling statistic from the AI deep dive.
The 136 drugs.
For 136 drugs, the analysis found that the 136 existing non-aging focused drugs may inadvertently accelerate biological aging.
And that finding just changes everything because those drugs were developed and approved using metrics that couldn't see this kind of subtle chronic molecular damage over time.
So, if we now have causality-enriched clocks designed specifically to detect genuine damage, the damage clock versus temporary adaptation, the adaptage clock, we had to ask the ultimate question.
We saw how tirzepatide prevented that detrimental metabolic adaptation.
Right.
Could that same protective mechanism extend to key biological aging pathways? Could these new drugs offer a systemic adaptation that delays overall damage accumulation?
Or could they carry their own subtle pro-aging signals that the old clocks just couldn't detect, and only the damaged clocks will reveal that, years from now?
It's a challenge of measurement that this next generation of clocks is uniquely positioned to answer, and it's something for you to mull over as this new era of pharmacology unfolds.